Hello and welcome back to the Computational Genomics Lab. We are doing GGPLOT II. Last time we got our basics down, and we're going to advance it a little more and make some new and different plots. I'm just going to load these packages here. Make sure you have them installed, including this broom package. It's kind of just makes your figures look a little cleaner. But how handy is this? We ran that NF core pipeline and we got our results, and we can just load them up in no time here. We're going to load the TPM results and our DESeq ResDF that we filtered our results by p-value and log fold change. And so we have now explored these 5 layers, and there's more detail again on each layer. And we're going to now look at multiple geometry layers, so where you can do multiple different plots on top of each other. Okay, so let's start with that. And so we can see here, again, ggplot is the function we're running to make the figures. Inside the parentheses, we take our filtered ResDF. That's our results data frame from DESeq, compiled with all the different comparisons and filtered for p less than 0.05 and log fold change greater than 2. Okay, so we're going to make x our base mean now. So we're going to say, okay, how abundant are you? And then the y-axis is your p adjusted value. So are the p values more significant at lower base means or higher base means or evenly distributed? We're going to find out in just a second here and have a new result. So those are our x and y, and then we're going to add geom point, and then xlim of 0 to 10,000, and a geom density now. So we're going to throw a density plot like a topo map of a mountain or thing like that. We can add that on top of our dot plot layer, and it's going to calculate how dense the points are in a given spot of the figure. Okay, well, cool. So we can see that most of our data is here in the bottom left corner, and so really what I think this is saying is that you're going to have a lower p-value the lower your base mean is, and that kind of makes sense from the way DESeq 2 works, is that if you're not very abundant, you're not going to you're going to have a little more statistical thresholding. And here we can see the topo map, or the density, that the peak is right down here, and then we have this long tail of base means out here where most of our data is here. And that tells you sort of in a statistical way of where the density of your data is. Okay, now that's geom density 2D. We made a density plot last time. That's different. That's just saying where your data density occurs across the x-axis. This is mapping it onto the actual dots and saying how dense they are there. Okay, so now if we do this, like sort of zoom in here, and right now we're doing geom point first and then geom density second, and that's the way we're doing it. That's fine, and we can see that the blue is on top of the black. If you switched them, they would go underneath. So the order does matter, basically. Okay, so now we're going to compare. The x-axis is log fold change, and now the y is going to be the standard error. So at that fold change, how much error had occurred in DESeq's analysis. Geom point, new x limbs of -5 to 5, because our x-axis is now log fold change, log 2 fold change, and geom densities on top of that. Okay, cool. So now we can see that most of our density is down here in the less than 2 to the negative 2.5 and 2 to the 2.5 is where most of our data is, and we have these density plots on top of the data. And you can see this one's much more dense right around 1, whereas this one's a little more diffuse and kind of leans into like 1.2. Just some aesthetic differences there in looking at up versus down. Now, what we saw there is, let's just change the order. So this is going to be the exact same plot, but we're going to put geom point on the bottom and density above it. So now we see that it's a little less useful this way, that the density is underneath the dots. So you're going to want to put the density last, so it plots on top. Okay, that's cool. We can add multiple layers of data onto the same plot, and we can fill by different aspects as you did last time. I encourage you to play around with adding a fill, color by in that AES layer. Okay, so what do you think the relationship is between p-value and p-adjusted? Well, we can plot that and start to find out. So we can say we're going to plot our data frame, our results data frame. X is going to be p-adjusted and Y is going to be p-value. And really, you can take any two columns in this data frame and see how they're related to each other. Okay, perhaps as expected, we would hope that p-adjusted and p-value follow similar trends. But you can see that there's almost like 4 lines here. And that's the way the p-adjust works. And we saw it in previous class. There's much more information on how that's calculated and considerations therein. But it's much better than just using p-value. And what you'll notice is that this slope is less than 1, in the sense that your p-adjusted should always be less than your p-value because it's being corrected. Sorry, a bigger number, less significant. Okay, so now we can add trend lines to this. So we could add our, like, is there an r squared here? What is the y equals mx plus b? And to do that, we're going to use the same starting point, x-axis p-adjusted, y p-value, a geom point like we have here. And now we're going to add this statistical layer, which is cool. So we're going to add a geom ab line. That's just going to make a line of whatever we tell it. And we're going to say, well, if they're perfectly linear related, the intercept would be 0 and the slope would be 1. And we're just going to make that. So that's not statistical. And then below it, we're going to add another layer of geom smooth method linear regression, or least squared. So there's a bunch of different methods here, and you can Google geom smooth and find your favorite way to do it. But what's nice about this is we can get some statistical analysis here. So if we zoom, so we can see that indeed a 1-to-1 relationship is not true, and that p-adjusted tends to be lower than, or a bigger number than the p-value. Okay, so and we see the blue line here is statistically saying where the trend line is in the data, and it looks pretty good, it looks pretty linear, but we don't have the information from that LM model. Sorry, it's a linear model that is fitting the data to a linear model. So that's pretty cool that in one line you can do that complicated statistical analysis. Okay, so let's do the same thing here, but we're going to retrieve the values. And so I believe this is just going to give us the exact same plot. Yep. Okay, so we just reran that. It's kind of redundant. But here we're going to make an object with LM fit. So we're going to get the slope and intercept for that blue line that R calculated fits the data. So if you had a hypothesis that it's linear, you can test that with a linear model and get determine if that's true. So this is, we're not doing ggplot now, we're just doing the LM function. Again, R is amazing for statistics, so it can just do it with 2 letters. We're going to take our res dfp adjusted and compare it with this tilde, means compare, to filtered res p-value, which are our axes here that made that line. Okay, so now we can see in our LM fit, it looks complicated, there's a bunch of values in here. So let's go do a summary. We haven't really done this, and we probably should have by now, but summary and table are great functions in R to just take a look at those tables and see what is going on. And so what we can see is that that's written out a little more clearly here, that our coefficients, our intercept is nearly 0, the error is very low, the significance is very significant, and we can see our R squared here is 0.965, so this is a pretty good linear fit based on those values. Okay, so let's go back and add this to our plot. And so, just to highlight how cool summary is we can also do it to summarize the base mean column in our res df. And what we get is the minimum, median, and maximum quartiles, and the average value in there. The number of, the average value in each of those bins. Same for p adjusted. We can see that the max highest number is about 0.05 where we cut it off, and the minimum is 0, which is great, very significant. So summary is really helpful to go into a table and just get the basic summary of that data. Okay, so we're doing the same thing here. We're making the LM, but the order does matter. So here previously we did p adjusted versus p value, and we're going to switch those around between these two sets of data. And I don't know if we're going to want to look at both of these summaries, but look for yourself. Go through here and look at LMFit and LMFit2 and do their summaries. And take a look in the console here, and you will see that the slope and intercept are all different. So you want to know what your first value is saying. How does p adjust relate to p value versus p value relate to p adjust? So they're kind of like 1 over each other, and so they're inversely related, but I just want to point that out. So we can take that LMFit data we have and use this Broom package, which we just installed, and make an object called LMDF, which is the data frame. It's just going to take that complicated table and make it nice and easy to read. So we can see here we get our intercept and significance, incredibly significant here. And yeah, just a much easier way to read that summary. Okay, so we're really doing the same thing, and we're going to compare now the summary of our model with them LMFit1, so this is really just LMFit2, okay, so we're going to plot this. So we're going to make a model, which is our LM for p adjusted versus p value, the summary, which we've just been doing. And now we're going to plot this again with the p adjusted and the p value, geom point, same thing. But now we're going to make a geom ab line with the actual data from that LM fit model. So we're going to say slope equals 1 over LMDF. In their column is an estimate, and it's the second one. We can see that in our console down here with that nice version of it here, that this is the second value in the table, and it's going to plot that and do a function in there of doing 1 divided by that estimate. Okay, so let's see how close our line now fits. Again, we're making it fit based on the LM, but instead of having R do the LM, we did the LM linear model and then plotted it. And we see pretty much the same thing. Nice fit here. Essentially is the same as the blue line because we put in its parameters. Okay, so we can look at some other geometry layers, like a bar plot is something that's very commonly used, and we can just say how many genes are there in each of the different time point comparisons or the result name. So if we run that, and we're using now the geom bar for bar plot, and we can zoom in, and again we see this same trend we've been seeing, that most of the genes are changing in the 0 versus 24 and 0 versus 48 time points. You can change the colors on these inside this geom bar. There's a lot you can do. And again, more detail is written down as I'm kind of cruising through this. Okay, so now what we're going to do is compare the x-axis is a bar plot, and y is going to be the base mean. So normally in a bar plot you wouldn't make the y-axis. You're not going to probably ever do this again, but it's good to know that you can actually tell GGPLOT what you want the y-axis to be instead of it calculating it for you. And so we're going to say the y-axis is equal to the base mean, and x is equal to the result name, like we did here. And we're going to say stat is equal to identity. And that's going to make sure that when we make the bar plot, that the y-axis is using an identity, which is this base mean here. Because normally when we made the bar plot, we don't have a y-axis. So now that we have one, we have to say what it's going to be. and that is the statistic identity, which is base mean. Okay, so there we go, and now we can see it looks a little different here. And what we see is that the base means seem to be going up as time goes on and then back down at 96 hours. So the base mean is, again, the abundance of each gene in each category of comparisons between the 0 time point. Okay, so we have, okay, a little more to go here. And what we're going to do now is we're going to take our filtered res df and we're going to make a sum of the base means. So we're going to pipe our data frame in there, and we're going to filter result name just to the 12 versus 0 time points. We're only going to look at that, and we're going to summarize the sum of the base mean equals the sum of base means. So we're telling it what it's going to be called, and we're going to take the sum of the base means in that time point. Okay, so now we run that. I don't have the package loaded. Okay, so that we can just ignore for now. And moving on. So now we are going to make some bells and whistles on our bar plot and color the bars and do some fun things to add where's the mean in each time point comparison, etc. So we're going to take our GGPLOT with the significant res df. X-axis equals result name, same as there. Y equals base mean, same as what we just did. This time we're going to make a geom point, and we're going to say statistical summary. So this is a ggplot function, which it can do a bunch of statistics for you. And the function we're going to run is mean. We're going to color the mean red. We're going to make it a shape of 1, which I believe is a triangle, and a size of 2. You can try out different numbers in the shape and find a shape you like. But I think you'll see why this is pretty useful. Is it saying that if we look here at all of the base means in each time point, that bar plot was really thrown off by that one dot up here, and that the mean is really way down here where that circle is. I guess 1 is a circle, 2 is probably a triangle, and it's circling where the mean would be in that distribution of base means for each time point comparison. Okay, so we have really gone through these 5 layers in a great deal, but we are going to kind of go back and look at the scale, which is kind of an aesthetic, like what colors do you like, things like that you can add. And again, there's a bunch of options here. So same plot. We're gonna do X as p-value, Y as p-adjusted. We're gonna color them if they're less than 0.01. Geom point. We're going to scale x-axis continuous. That's just a function I'm throwing in there so you have an idea of what that means. It's going to try and make the x-axis fit the data the best. And then here's where the scale color brewer is cool. We're going to say palette equals set 1. And you can Google, again, scale color brewer, different palettes, and there's a whole website of different colors that you can pick your favorite. And when we do this, we can see that as we hope, all of our p less than 0.01 are in that 0.01 range. So we're just highlighting those values, and the distribution of p-values versus p-adjusted. We saw this same plot flipped over previously. But just that you can start to color them. And now we can change the same, we're going to do the same thing, but make really significant p less than 0.025. Same thing, adding limits. So we're just kind of adding in all the things we've just done and add it all together. And we can see now that only those very first dots are colored to the 0.0025 or whatever we cut it off on. So this way you can just highlight where your significant genes are relative to some threshold of, in this case, 0.0025. All right, so you are now hopefully getting a lot of the basics in ggplot. Go back through, there's a lot there. I went through that kind of fast. and only really pick out the things you think would be useful to you. We're really just kind of going through and providing a menu, but in the future you'll decide what you like, and we'll see you next time for GGPLOT 3.